Hello, and welcome to Chapter 10. Your Media Under the Microscope article in Chapter 10 talks about this phenomenon called chimerism. Chimerism is actually where cells within a single individual can express two different genotypes. And oftentimes we see this bisymmetry that goes on in true chimerics. What we oftentimes think is that the chimeric has actually absorbed a twin, and therefore some of the cells exhibit one genetic profile while other cells exhibit the other genetic profile. So we're going to talk a little bit about genetic engineering here in this chapter. And basic science of genetics, of genetic engineering, there are really no products or applications that are directly derived from the basic science. We just simply look at how the microorganisms duplicate, how the genetic information is exchanged, and how they use their genetic information. Applied science of genetic engineering, however, shows applications from the basic research science. So for examples, genetic manipulation of the transfer of genes between a farmer's two largest pigs so that only advantageous genes are selected so their offspring will also be large. Utilizing DNA to identify suspects in crimes, and fixing the underlying genetic mutations to treat diseases can all be examples of the applied science. We can also utilize RNA regulatory molecules to permanently fix diseases. So the intrinsic properties of DNA. Helicase, the enzyme that unzips the DNA and forms a replication fork, is able to unwind the strands of DNA in the double helix just as easily in the laboratory as it does in the cell. DNA strands separate also when exposed to temperatures just below boiling. Complementary strands will hydrogen bond with the original strands, and strands will reform when the DNA then is cooled. So, what we'll see is that the DNA responds to heating by denaturing, basically separating the two strands, losing the hydrogen bonds between the nitrogenous bases. When cooled, the two strands rejoin at the complementary regions. The strands need not be from the same organism as long as they have matching nucleotides. Restriction endonucleases. This is a group of enzymes that actually cuts DNA crosswise at selected positions. They can recognize foreign DNA that has been put into a piece of genetic code, and they are capable of breaking the phosphodiester bonds between adjacent nucleotides on both strands of the DNA. They are used to protect bacteria and archaea from bacteriophage or plasmid DNAs. Each endonuclease recognizes a sequence of around 4 to 10 base pairs. They can recognize and clip the DNA at palindromes, those inverted repeats, those sequences of DNA that are identical when read from the 5' to 3' direction on one strand and the 5' to 3' direction on the opposing strand. They can cut either forming sticky ends where there's staggered symmetrical coats that leave short tails of unbound nitrogenous bases, which usually we have about 4 to 5 bases on each sticky end. And the base pairs with complementary sticky ends on the other DNA fragments or plasmids then can actually slip into these sticky ends. Or they can actually cut at what we call blunt ends, basically just simply cutting across both sides of the DNA equally. So this actually shows some of these examples of these endonucleases, and I won't get into the different types, but for example, the Echo RI actually cuts right here, and again, this is GAATTC, reading in the opposite direction, GAATTC, and so we wind up with about 4 bases of a sticky end. The HIN D3 will actually also cut a sticky end, and the HEI3 will cut a blunt end cut. When we cut sticky ends especially, as long as we have an opposing sticky end from another piece of DNA, then we can just simply splice them together. So for example, if we cut an organism's DNA and we cut out our selected piece of DNA and we use like a plasmid here or DNA from another organism and it had the corresponding bases on the sticky ends, then they simply will come together very easily. With blunt ends, we have to be a little more specific and use DNA ligase. DNA ligase will actually help to form all of these, whether they're sticky ends or blunt end cuts. What is cut is the restriction fragment, and this is the piece of DNA that's produced by the restriction endonucleases. And sometimes we refer to these as restriction fragment length polymorphisms. It's the differences in the cutting pattern of specific restriction endonucleases that give rise to the restriction patterns of different lengths. This will allow for direct comparison of DNA of two different organisms at a specific site. DNA ligase will be necessary to seal the sticky ends or put the blunt ends together. The main application of ligase is the final splicing of the genes into plasmids or into a second organism's chromosomes. Reverse transcriptase is another enzyme that we use. This is actually seen in the replication of HIV and other retroviruses, and in fact we borrow this from these viruses. It is able to convert the viral RNA into a complementary DNA, and the reverse transcriptase will do this in either the host cell or in the laboratory. So we can take a piece of RNA and make a complementary DNA strand from it. And the reverse transcriptase doesn't really care what the RNA source is, so we can use pretty much any form of RNA that we want to make complementary DNA. This is extremely useful in synthesizing eukaryotic genes from messenger RNA transcripts. Because remember, since we have to modify the messenger RNA in eukaryotic cells before it goes for translation, we have all of those introns, the non-coding portions, that we have to take out first. Therefore, when we utilize the mature messenger RNA of the eukaryotic cell, the gene is then free of the introns. So we simply will then allow the cell to take out the introns and splice together all of the exons, and then we can extract that mature messenger RNA, put it in with a reverse transcriptase, and allow it to make our complementary DNA strand. This double-stranded complementary DNA then contains only the coding portions of the eukaryotic DNA. You may have heard of CRISPRs. CRISPR are clustered regulatory interspaced short palindromic repeats. A lot of words in one little acronym. In bacteria or archaea, CRISPR genes actually will cut the DNA with repeating nucleotides. The enzyme in the CRISPR system will recognize and cut out foreign DNA left behind by invading bacteriophages or plasmids from other bacteria that is not useful to the cell. Scientists then exploit these enzymes within the CRISPR system to cut DNA in just about any organism exactly where they want to. Now then, how do we analyze the DNA? Well, oftentimes after cutting it or making complementary pieces of it, we can then go put it into gel electrophoresis. This produces a readable pattern of the DNA fragments based upon weight and electrical charge. Samples of DNA are placed in compartments called the wells in a soft agar gel and subjected to an electrical current. The negative charge on the phosphate ends of the groups cause the DNA to move towards the positive pole on the gel, and the rate of movement of the DNA fragments through the gel then is based upon the size of the fragment. The larger the fragment, the closer it remains to the wells. Positions of the DNA fragments then are determined by staining the DNA fragments within the electrophoresis gel, and these patterns are actually very distinctive. It's useful in characterizing the DNA fragments because the heavier ones stay closer to the wells, lighter ones move farther away. It allows for comparison of genetic similarities among samples in genetic fingerprinting. So, for example, we take a DNA and we use basically 3 restriction endonucleases, and it cuts the DNA into 4 segments or restriction fragments. We then put the DNA into these wells and then run it through the electrical charge. We typically will also use something called the ladder, and this actually has known fragments of the DNA based upon molecular size. So we can use the ladder as a comparison for the ones that we are looking at. And so that we see that in well 3 here, we actually have 1, 2, 3, 4 fragments that have been separated. And in the other wells, we have 1, 2, 1, 2, 1, 2. So we know that in well 3, this was the fragment that was cut by these 3 endonucleases. 2 different nucleic acids can hybridize by uniting at the complementary regions if they share any in common. Single-stranded DNA can hybridize with another piece of single-stranded DNA or single-stranded RNA. RNA, however, can hybridize only with another piece of RNA. Because of this hybridization, we can actually make gene probes. These are short stretches of DNA of a known sequence that will base pair with a stretch of DNA with complementary stretch if the DNA exists in a sample. The carrier probe usually will then be processed with either a fluorescent dye so that if hybridization does occur, it can be visualized, or some other sort of marker. Enzyme-linked probes are detected when non-pigmented substances become colored when hybridization occurs. Use of gene probes then allow for diagnosis of the cause of infection from a patient's specimen, because if we get hybridization of the known probe with the sample from the patient, we can then identify what the disease is caused by. We can identify a culture of an unknown bacterium or virus. So this is what we can do. We can actually make a master plate with colonies of an unknown bacteria. We then will cover that plate with a filter membrane, picking up pieces of each of the colonies. This then will treat the filter paper with a dye to lyse the bacteria. And so we have strands of bacterial DNA, and we can use then different mechanisms to cut that DNA. We treat the filter paper usually with sodium hydroxide to separate the DNA into single strands. Then we add known probes onto the filter paper. If the probe will hybridize with a piece of the bacterial DNA, then we know that that particular unknown sample is going to be what we're looking for. And then we can take that filter paper where we see the fluorescence and actually pick up those colonies from our original master plate based upon their location on the filter paper. The nice thing about nucleic acid hybridization tests with these gene probes is it does not require electrophoresis. The DNA is isolated, denatured, and placed on the absorbent filter and combined with a microbe-specific probe, and then blot is then developed and observed for areas of hybridization, and then when we can identify which specific one of these colonies is our target bacterium. Fluorescent in situ hybridization, also known as FISH, the probes are applied to intact cells and then are observed microscopically for the presence and location of specific genetic marker sequences on the genes. This is a very effective way to identify specific genes on a chromosome. It's also effective in identifying bacteria living in natural habitats without having to culture them. And we can even use fish to detect DNA in cells and in tissues. So this is what we do. We heat up the DNA, and then we put in a probe that has a fluorescent label. If hybridization occurs, then the fluorescent dye will actually glow, and we will be able to see that. So here we've actually got some hybridization that has occurred within these pieces of our chromosomes here, and these are seen by the fluorescence that occurs. And we have both red and green fluorescent dye labels, and so these are different ones. The relative size of nucleic acids are denoted by the number of base pairs, A, T, C's, and G pairs, that they contain. Palindromic sequences, I said, were recognized by restriction endonucleases, and typically these restriction sequences are about 4 to 10 base pairs in length. The average gene in an E. coli bacterium is about 1,300 base pairs, or 1.3 kilobases. The entire genome of E. coli is about 4,700,000 base pairs, 4,700 kilobase pairs, or we could even go 4.7 megabases. The Epstein-Barr virus's entire genome is only made up of 172 kilobases, or 172,000 base pairs, whereas the human genome is about 3.1 to 3.2 billion base pairs. Now, we've mentioned polymerase chain reaction, or PCR, and PCR has become a real workhorse in the study of genetics. PCR is really a molecular Xerox machine. It rapidly increases the amount of DNA in a sample without the need for making cultures or carrying out complex purification techniques. It can replicate a target piece of DNA from just a few copies, even one copy, to billions of copies within a very short period of time. The sensitivity, it can detect cancer from a single cell. So all we need is one copy of the mutated gene. It can diagnose an infection from a single gene. PCR uses the same events of DNA replication. We're going to open the double helix, basically denaturing the double helix by using heat. We then use the exposed strands as templates. We use the addition of specific RNA primers that will bind to the DNA at specific sites. Then we use the action of a heat-temperate DNA polymerase. And so the specialized ingredients that we use in PCR, the RNA primers, we actually can make these so that we know that they're going to bind at specific sites on the denatured DNA. So they're synthetic oligonucleotides of a known sequence of about 15 to 30 base pairs of RNA that indicate where the amplification process should begin. We're going to use a heat-stable DNA polymerase from a thermophilic organism, and so sometimes this DNA polymerase is known as Taq polymerase because it comes from a heat-resistant microorganism. Since high temperatures just below boiling are necessitated or needed for dividing the DNA strands, then we have to have this heat-resistant polymerase. PCR technique then utilizes a thermocycler that automatically performs a cyclic temperature change. And there's 3 basic steps: denaturing under the heating, priming with the addition of the RNA primers onto the DNA, and then extension, allowing the Taq DNA polymerase to then add nucleic bases in the 5' to 3' direction. As it cools, the copied DNA will adhere, and then we reheat it, separate the newly synthesized strands, and repeat this process over and over. So, starting materials: the Taq polymerase, the RNA primers, and the DNA nucleotides. So here's our piece of DNA, and this is our targeted sequence. So in cycle 1, we denature by heating to 94 degrees Celsius to separate the two strands completely from each other. The primers then bind to the DNA at specific areas on both sides where the replication will start. Then extension occurs as the temperature then begins to drop to about 72 degrees Celsius. The Taq polymerase binds at the primers on both sides and will then lay down new DNA molecules along the entire length of each of the separated original strands. Then we heat back up and separate these two strands and make copies of those. And this just simply repeats and repeats and repeats until finally we are left with sometimes billions of copies of the target DNA only. Real-time PCR can detect the products during the reaction instead of waiting till the end of all the cycles and then figuring out how much we have. PCR can also be adapted to analyze RNA strands. RNA will be converted to DNA with reverse transcriptase, and that complementary DNA is then copied in the PCR reaction. PCR plays an essential role in gene mapping, the study of genetic defects and cancer, forensic science, and infectious disease diagnosis, and even plays a role in taxonomy studies. And in fact, some organisms have had to be reclassified based upon the genetic studies that use PCR to multiply the copy of target DNA by many, many times. So, recombinant DNA technology. This is deliberately removing genetic material from one microorganism and combining that with a different organism. This will form then genetic clones. Cloning is actually the removal of a selective gene from an animal, plant, or microorganism, which is then referred to as the genetic donor. It is then propagation of that gene in a different host organism that results in the genetic clones. The gene is normally inserted into a vector, either a plasmid or virus, that will then be inserted into a cloning host, which is usually either a bacterium or a yeast. Strategies for obtaining genes in isolated states include DNA that's removed from cells and separated into fragments by endonucleases, or genes can actually be synthesized from isolated mature messenger RNA transcripts using reverse transcriptase and creating the complementary DNA. A gene can also be amplified then using a PCR. Genomic libraries, these genes are maintained in the cloning host and vector just like in microbial pure cultures. And these genomic libraries then are collections of this complementary DNA clones that represent the entire genome of numerous organisms. So, a genomic library is stored as a set of bacteria, each carrying a different fragment of human DNA. And for simplicity in this picture, only a few representative fragments that are colored are shown. But in reality, we actually will copy all of the DNA fragments and put them into individual clones. So we'll take the piece of DNA, we'll cleave it with restriction endonucleases. So we cut up the chromosome. Then we can actually take those genetic fragments, insert them into plasmids, and then introduce those plasmids into bacteria. And so you'll see this particular bacteria now has the green gene, if you will. And all subsequent generations of that particular bacterium will carry that green gene along with the rest of the chromosome. Same thing for the red, the blue, the purple, and the yellow. Now, cloning vectors include plasmids, and because they are small, well characterized, and easy to manipulate, they make a very nice cloning vector. They can be transferred into the appropriate host cell through a artificial form of transformation. Plasmids that carry genetic markers for resistance to antibiotics are often used in order to study resistance. The other major cloning vector is going to be a bacteriophage. They have the natural ability to inject the DNA into the bacterial host through transduction. We can actually choose what DNA the bacteriophage will carry, and therefore the bacteriophage then will vector it through transduction into the host bacterium. Important aspects of these cloning vectors, usually we have an origin of replication, or an ORI, so that it will be replicated by the DNA polymerase of the cloning host intact. The cloning vector must accept the DNA of a desired size. So if we want to have so many base pairs, we have to pick a vector that is able to actually handle that particular size of our DNA that we want to clone. So desirable features of a microbial cloning host. The host has to have a rapid turnover. They can usually be grown in large quantities using ordinary culturing methods. They typically need to be non-pathogenic. We need to know their genome and have it well delineated and mapped. And the cloning host must be able to accept the plasmid or bacteriophage vector. And they'll need to be able to maintain the foreign host through multiple generations and secrete a high yield of the protein expressed from those foreign genes. So, what you need for gene cloning is the gene that you are interested in. Example, a gene for production of an interferon. A cloning vector, and again, this could be a plasmid or a bacteriophage, depending upon size. And then we simply will put that plasmid into the host. So we isolate the gene, we put it into the plasmid using restriction endonucleases usually to actually splice that gene into the plasmid. And we then can introduce that recombined plasmid into a host cell. That host then will be able to incorporate that foreign DNA and read the genetic code. Now, this occurs or can occur actually in the cell simply by, again, taking a piece of that cloned vector donor gene, making transcription to messenger RNA, and using the cell's protein synthesis to make the protein product. We can then actually harvest that protein product and purify the protein. Or we can simply recombine the bacteria, let them multiply in the media, and make lots and lots and lots of this protein and purify it. So as the cells multiply, the plasmid replicates along with the cell's chromosome. So in just a few hours of growth, there can be billions of cells, each containing the foreign or donor gene. Once the gene has been successfully cloned and tested, this step does not have to be repeated. The recombinant strain can be maintained in pure culture for production purposes. So we need to make sure that the host cell can actually read the donor DNA and make the appropriate protein, in this case an interferon. So DNA can be cut into smaller pieces by using enzymes called what? Complementary DNA is produced by using the enzyme what? And true or false, PCR uses specialized DNA polymerases adapted to high temperatures. And list and describe the two cloning vectors and why they are good cloning vectors and why they are good for the hosts. We can use recombinant DNA technology to then make these recombinant organisms. For example, most human insulin is now actually produced by recombined E. coli bacteria. They can be then the source of these protein products so that we do not have to rely upon eukaryotic cells to make human insulin, but we can actually use E. coli to make lots and lots of the human insulin for us. We can use recombinant DNA to actually study the nucleotide sequence of these genes, and we can even make medications that cannot be manufactured by other means. Oftentimes, we're using recombinant organisms now for large-scale manufacture of hormones or enzymes. And this table is not an all-inclusive table of recombinant DNA technology products, but certainly interferons, interleukins, which are type of cytokines that regulate immune function, and tumor necrosis factor that can be used to treat cancers. Erythropoietin, which is a normal enzyme hormone in the body that stimulates bone marrow to actually make red blood cells. And the erythropoietin can actually be used to treat some forms of anemia. Human growth hormone that can stimulate growth in children with pituitary dwarfism or prevent wasting syndrome. Certain enzymes can be made, such as tissue plasminogen activating factor or TPA. TPA can actually dissolve potentially dangerous blood clots in things like heart attacks or strokes. And certainly vaccines. Vaccines for hepatitis B, the human papillomavirus, and Haemophilus influenza type B meningitis are actually produced by organisms that have been recombined. And then miscellaneous things like factor VIII, which are needed in hemophiliacs to replace the missing factor VIII in their blood clotting mechanism, or bovine growth hormone given to cows to increase both milk and meat production. Or again, certainly go back up here to hormones, human insulin. Transgenic organisms are recombined organisms that are produced through the induction of foreign genes. They are also known as genetically modified organism. And if you look at a tomato here that's a heritage tomato versus a GMO tomato, what's the difference? And just looking at them, you can see that certainly a size is a difference. So we can genetically modify the tomato to grow a larger fruit. Bioremediation can also use recombinant microbes. Several types of naturally occurring bacteria can actually clean up oil spills or degrade pesticides. But we can actually take the genes of those bacteria and put them into other bacteria that replicate at a higher rate and actually create these recombinant bacteria that now can eat up the oil or degrade the pesticides or other toxic substances in the environment. Other uses include preventing ice crystals from forming around plants or actually even inside plants. They can destroy invading insects so that we get a higher crop yield. Or they can be used in the production of biofuels. And we could even produce new drugs with novel properties. And this has been a big area of study in antimicrobial drugs. And we can produce industrial chemicals in a more environmentally friendly manner. Controversy, however, surrounds the release of genetically engineered organisms, and certainly in genetically engineered plants. There's a concern that the genetically modified plants will share their genes for herbicide, pesticide, and virus resistance with natural plants that lead to something known as superweeds that nothing would kill. Scientists counter that non-genetically engineered plants and organisms swap genes all the time, and that inserting an insect-resistant gene into crops can actually reduce insecticide use on those crops. Several hundred strains of transgenic animals have now been introduced by research and industry. Unlike bacteria and yeasts, animals can express human genes in organs and organ systems that are then very similar to those of humans. Animal models have been designed to study then human genetic diseases. These natural systems are then used to test new genetic therapies before they are used in humans. Sheep and goats especially have been engineered to become factories for many of these protein products. And in fact, sheep and goats now manufacture many human proteins and excrete them in their milk or semen. And this process of creating these transgenic animals to produce proteins for human consumption then is known as farming with a PH. So, an expression vector carrying a desired gene can be put into an animal egg, which is then implanted into a surrogate mother. The transgenic offspring then produce the new protein in their milk or semen, and the milk is easily harvested and the protein isolated, purified, and then made clinically available for patient use. So, can we create a living organism from non-living things? Well, I actually thought we put that to bed when we talked about spontaneous generation and biogenesis. However, it's come back around. Synthetic biology. This is the creation of organisms from chemical scratch. Craig Ventnor back in 2010 actually created a self-replicating bacterium using 4 nucleotides of DNA. This was the first time a living replicating cell had been created from the raw chemicals. Scientists could potentially then create precise chemicals to replace those missing in disease processes, assemble customized immune components, or construct biological molecules that can precisely target cancerous cells or pathogenic microbes. Surprising results of synthetic biology is actually been also the creation of what we call xenonucleic acids, or XNA. These contain different sugars than DNA or RNA, or in some cases actually no sugars at all. Enzymes were able to copy these XNAs, resulting in the ability to store information and pass it on to descendant cells. These findings imply that life on other planets does or did not necessarily require DNA or RNA to exist. But ethical questions are brought up by research into synthetic biology. There are fears that whatever we create could get away from our control and have unintended consequences. The potential benefit of being able to design cells that create biofuels or medical or agricultural products may, however, eventually outweigh the hypothetical concerns. So, can you list and describe 3 products produced through recombinant DNA technology? Pseudomonas syringae has been modified to prevent the formation of what on plants, and you may have to do some research on that one. Transgenic sheep and goats produce human proteins in what? And in 2010, Craig Ventnor produced what using 4 nucleotides of DNA? So, one big advantage of genetic recombination is gene therapy. And for certain diseases, the disease phenotype is due to the lack of a single specific gene and protein product. For example, type 1 diabetes is caused by the lack of insulin because the individual does not have the gene to produce the insulin. Insulin was first provided to treat these patients from the pancreas of pigs or cows, but this was a very time-consuming process. So recombinant human insulin was the first genetically engineered drug approved for use in humans. And now all of the human-based insulins that we use in both humans and animals today are all recombinant products. And it's primarily from recombinant E. coli. So gene therapy is the concept to repair or correct the faulty gene in humans suffering from a fatal or debilitating disease. The benefit of gene therapy is it would provide a permanent cure of the physical dysfunction by repairing the genetic defect. Strategies for gene therapy is that the normal gene is cloned in either retrovirus or adenovirus vectors. Tissues are removed from the patient and are incubated with the genetically modified viruses to transfect them with the normal gene. In other words, the normal gene is now part of the viral genome. The transfected cells are then reintroduced into the patient's body. We could also use naked DNA or a virus vector to directly introduce these genes into the patient's tissues. So this is what we see. So a normal gene is isolated from a healthy subject. The gene then is cloned, usually through PCR. The gene is then inserted into the retrovirus vector or adenovirus. A bone marrow sample is taken from the patient with the genetic defect. The marrow cells are then infected with the retrovirus. These transfected bone marrow cells then are reinfused into the patient, and then the patient is observed for expression of the normal gene. So what the retrovirus or adenovirus is carrying is only the gene that we want to replace. It is not carrying viral genes. So what we're hoping is that when we put it back into the patient, that these now transfected marrow cells will then produce the product that is the normal product of the normal gene. Experimentation and clinical trials of gene testing have been carried out on human volunteers with the particular genetic condition. Most gene therapies target cancer cells, single gene defects, or infections caused by genetic dysfunction. Most gene deliveries are carried out by virus vectors. Early trials were hampered by safety issues in which the viruses used as delivery vehicles actually caused malignancies in the cells in which they were injected. Germline gene therapy would be the absolute best, and this is where the normal genes would be inserted into the egg, the sperm, or the early embryo, so that subsequent generations of these cells would have the corrected gene. This means that the therapeutic gene is now inheritable. Diseases that result from inappropriate expression of proteins include Alzheimer's disease, certain viral diseases, many cancers, and sometimes this inappropriate expression occurs due to a malfunctioning microRNA or another regulatory RNA. Cells then use a variety of these microRNAs to actually silence gene expression in normal cells. So we could actually use the microRNAs to fight diseases. Introducing specific microRNAs into small cell lung cancers in mice actually blocked and stopped the tumor of the cells. MicroRNAs cause regression of liver cancers in mice. And we could even use these microRNAs to fight certain diseases. Inhibiting specific microRNAs in mice actually stopped the growth of ovarian and breast cancer in the mice. And an inhibiting microRNA in African green monkeys actually lowered their plasma cholesterol levels. Multiplication of hepatitis C in chimpanzees was inhibited by shutting down one of the classes of microRNAs within their cells. Now, the first successful human application of interfering RNAs was done with the respiratory syncytial virus. Healthy human volunteers were given a nasal spray containing a microRNA designed to silence a gene from the respiratory syncytial virus. Volunteers were then infected with RSV and used the spray containing the microRNA for 5 days. 44% of those using the microRNA that were infected became infected, compared to 71% of those that used a spray that did not contain the microRNA or the control subjects. And of those 44 that did become infected with RSV, their symptoms were significantly reduced. So, a concept check. Can you name the 2 viruses that are primarily used for gene therapy? True or false? Naked DNA is often used in gene therapies. And true or false? There have been no successful human trials of gene therapy. And what type of therapy describes gene therapy that's introduced into effective egg, sperm, or embryo? And gene expression can be silenced using what? Now, analysis of DNA can give more accurate mechanisms of differentiating among organisms. It can identify an organism that's no longer present, but we could find the traces of DNA that were left behind. Example, when a criminal is identified by DNA extracted from hair left at a crime scene. Or how about a disease that has left part of its DNA behind, but the organism is now no longer present? We could analyze DNA that can identify a person at increased risk for cancers or Alzheimer's disease before symptoms actually arise. Sequence maps are the most detailed maps of a particular genome. It gives an exact order of the base pairs in a plasmid chromosome or the entire genome itself. Genomes of thousands of organisms have now been sequenced since we finally broke through with the human genome in the year 2000. And this includes viruses, bacteria, and eukaryotic organisms, including us. It's interestingly enough that when we look at the sequence maps of humans, that we share approximately 95% of our DNA in common with the great apes, 80% with mice, about 60% of our DNA is shared in common with rice, and even 30% of our DNA is actually shared with the worm C. elegans, which is one of our roundworms. How we do genomic mapping sometimes is through whole genome shotgun sequencing. The whole genome of an organism is broken down into smaller manageable fragments, usually by, again, these endonucleases. The fragments are then separated through gel electrophoresis, and each fragment then is inserted into a plasmid and cloned in a bacterial cell, primarily E. coli. The Then the plasmids are purified and the DNA fragments are sequenced by automated sequencers. And a computer program then takes all the sequence data and is able to find where the sequences overlap and produce the entire picture. Contigs are larger contiguous sets of nucleotide sequences that are produced by the automated process, and then the ends are compared and see where they overlap. So contigs then will be put into the proper order to determine the entire sequence. And then finally, a human does actually do editing to examine the sequence and their overlaps, looking for any irregularities, frame shifts, or ambiguities. So we'll take the entire chromosome, chop it up into pieces, put it into the electrophoresis, and then take those individual pieces and actually then make these sequences and then clone them in a clonal host. And then we look for overlaps in the plasmids that are taken from these clonal hosts. And then the human analyzes the edited computer output to make sure that there are no errors in the overlaps. High throughput sequencing is another way of gene mapping, and this is where sophisticated computers and systems were developed to sequence genomes in a shorter period of time with less human effort than the whole shotgun approach. So we take the DNA isolated from an organism. Again, we fragment it using endonucleases, and then adapters are added to those segments. The sequences then undergo PCR, where they are immobilized using the adapter sequence and copied via the PCR process. Millions of amplified sequences then are sequenced inside the machine using various methods looking for overlap and other sequences. And the sequences then are aligned using software, and the entire genomes then can be revealed. Genomics and bioinformatics. Genomics can be managed or manage the data produced through these high throughput screenings. We can analyze and classify genes, we can determine certain protein sequences, and ultimately determine the function of the genes. Now, we have mapped the human genome. meaning we know the entire sequence of base pairs within the human genome. Do we know what all of the genes do yet? And the answer is no, because we've not completely done the entire processing of classifying the proteins produced by every gene and what the functions of those proteins are. But we have actually mapped many of these. In fact, on chromosome number 16, I think we are now at the point where we know what all the genes are and what their protein products are from each gene on chromosome number 16. And we have mapped the majority of many of the other genes, but we're not quite fully done even with the human genome. Annotating the genome simply means determining the functional information of the gene sequence. Well-annotated genomes will provide a complete understanding of how the cell functions normally, how diseases develop, and the process of aging. It will also characterize genetic mechanisms behind pathogens and possibly develop new treatments. DNA profiling, also called DNA typing or DNA fingerprinting, shows us a unique picture of the genome. It's a tool of forensic science that was actually developed as early as the 1980s. It's based on the principle that the way the nucleotides are combined is different for every organism, and it is used to differentiate between organisms causing disease outbreaks or criminals. So we can take the DNA from various organisms. We can use endonucleases to cut them and then put them through gel electrophoresis. And then we can even add probes to the fragments in order to be able to visualize the bands. We can then compare organisms to organisms. So for example, if you look at organism number 1 and organism number 4, they share in common some actual sequences. So they share this sequence in common, they share this sequence in common, and they share this sequence in common. That means 1 and 4 are related, but are they identical? Well, they're not. If we look at sample or organism 1 and 2, they again share a couple of sequences in common, and samples 2 and 3 also share some commonalities. And in fact, actually, 2 and 3 are closely related, and 1 and 4 are closely related. While 1 and 2 and 3 and 4 are all related, 1 and 4 are more distantly related to 2 and 3, and vice versa. So if we look at this profile down here, in lanes 1, 5, and 9, we have known fragment segments, and these are our ladders or markers. We also have another control here in lane 8, so we won't look at those. What we're looking at is DNA from the victim, which is in lane 2, DNA from evidence that's taken from two different sources here. So evidence 1 and evidence 2. In fact, these were actually semen samples taken from the rape victim. And then we have two suspect DNA profiles. So I have suspect 1 here and suspect 2 here. So which one are you going to arrest for the rape? Restriction fragment length polymorphisms, where these restriction enzymes cut DNA at the specific palindrome recognized sites. So if a given strand of DNA possesses the recognition site, the restriction enzyme will cut the DNA at that point. If the same strand of DNA is from a different person and does not have that restriction site, then the restriction enzyme will not cut the DNA and it will remain a larger piece of DNA. Each of the two samples then is digested with restriction endonucleases and separated on an electrophoretic gel. The first will display 2 bands, while the other one will only display 1 large band. All method of DNA fingerprinting depends on the same variation of this strategy. Now, single nucleotide polymorphism, or what's known as SNPs. These are important in individual traits. In SNPs, only a single nucleotide is altered within the genes. However, tens of thousands of these SNPs actually can exist at a single locus and exist throughout the genome. Scientists are now mapping these SNPs to determine which ones put humans at greater risk for particular diseases. So, for example, in our current coronavirus outbreak, it has actually been shown that some people actually simply are immune to coronaviruses. And is this due to the fact that they have SNPs within their genome that make them at less risk of actually contracting coronaviruses? And that's a question we need to answer. Many companies will map SNPs and give people an estimated risk for a variety of disease based upon their single nucleotide polymorphism. So, for example, if in our amino acid sequence here at 506, and this is the position, the number of the amino acid, if we have arginine at this point, we actually have a protein clotting factor 5 that is inhibited by another protein binding at the amino acid 506 of arginine, and we actually then will get normal clotting. However, if we have a SNP in this gene, so in other words, instead of GCT, we now have GTT that codes for the glutamic acid here, or I'm sorry, glutamine here, then this protein factor will not be inhibited and we actually get excessive clotting. Now, microarrays allow biologists to view the expression of genes in any given cell. They track the expression of thousands of genes all at the same time. And so we use again DNA clones, and we can take the DNA, hybridize it, and put it in these microarrays and actually then see where hybridization occurs. Most of you will never ever see a microarray, so I'm really not going to spend a lot of time with it. But we can use them to develop sensitive diagnostic tests that will search for specific pattern of gene expression and be able to identify even a subtype of cancer, allowing doctors and pharmacists to treat each cancer with drugs that will be most effective for each type of cancer that can be seen in a single person. So, the last concept check. Describe the steps involved in whole genome shotgun sequencing. What genomes provide a complete understanding of cell function, disease development, and other issues? DNA profiling depends really on what type of enzymes. And lastly, what allows scientists to view gene expression in any given cell? So this is the last of this chapter, so we'll see you in the next one.